Well, we're starting to realize that the transcriptome doesn't always predict what the actual cell will do. More important also is the epigenome above that level, and together dictating what the proteome is, the actual actors in the cell. And you get these, and you can start making these pathways. Now, these are a little bit nonsensical, I think, and I try to equate them to what Delta Airlines flight plan might look like. And we actually, I have a friend of mine who calls these the reticulum, because we don't really understand what these mean. We're starting to understand what the hubs are. But next we're going to have to profile even more intelligently. We're going to have to go up to the metabolome at the single cell level. And we're going to have to go up to the connectome, and ultimately the organoid. And that's what we're going to start looking at in a dish. For example, for the nervous system, that was something that a postdoc in the lab there, he seemed to wanted to do. He wanted to recapitulate neural tube formation. And through a lot of steps that I can't go into, was in fact able to get neural tubes in a dish that he could then begin to understand, profile, and test. Going in to an injury. Ted Tang, who's now an associate professor at Harvard, but when he was a postdoc in my lab, in collaboration in work we did with Bob Langer at MIT, we wanted to recreate abnormal sections in the spinal cord. There we realized we needed a bioengineering solution. We needed an organoid, so to speak, even back then. And what we would do is create cells on a scaffold, and the function of the scaffold was designed so that it emulated the outer segment of the spinal cord, the white matter, and an inner segment of the scaffold that emulated the inner segment of the spinal cord. And then together, put that in there and allow a return of function, because now axons could bridge the gap. Interestingly, they were host axons. In a stroke, same kind of organoid kind of solution. In a stroke-like lesion, cells on a scaffold that now would reorganize themselves. Here's the scaffold, and here are neural elements that are now filling in a gap that then get vascularized and together start creating. Here's where the gap would normally have been in this stroked out animal, now creating this kind. Is it beautiful, textbook, well laminated? You'd be proud to show your neurobiology teacher type parenchyma? Absolutely not, nothing like it. But is it a way of starting to reconstruct the circuitry so that the animal can do better and do it spontaneously in the right ratios? That is what that did show. So now we're going to start looking at organoids. That's what we're going to have to profile when we start predicting. And then we have to start making sure we're looking at assays that are clinically meaningful. So what does that mean? Well, a whole area of stem cell biology is starting to model diseases in a dish. And in fact, it's called disease in a dish. Now, one way to do that is to take normal cells and assault them in ways that you know, perhaps, or you at least think that a disease injures cells. Another is to take cells, skin cells, blood cells, from a patient who's actually diseased, turn them into stem cells, and then try to figure out its fingerprint, the mechanism of disease progression, what the prognostic, diagnostic, the target, and then discover the drug against that target. So again, you've heard Daniel talk about that, but taking now skin cells or blood cells from this patient, put it back in developmental time, and now you're able to kind of interrogate cells from living patients and all their family members in a way that previously was inaccessible. So that works very nicely with monogenic diseases. These are neurons from a Rett syndrome kid. So presumably these neurons have Rett syndrome. The real challenge over the next 5 to 10 years is how do we model diseases where we don't know the gene, we don't know the pathway, we don't know the cell type even involved, we don't know the region of the brain really involved. That is going to be the real challenge, modeling complex, polygenic, multifactorial diseases. And we've kind of embraced that challenge, and neuropsychiatric disorders are the poster child for that. Very, very poorly understood. So not only are they a critical entity in themselves, but if we could figure out how to tackle these as proof of concept, then it really would advance the field of disease modeling, which is going to be very important over the next 5 to 10 years. So we devised for one of these something we call the molecular can opener strategy. And that's making iPS cells from a patient, and the can, of course, is going to be these complex, poorly understood diseases, of which neuropsychiatric diseases, I said, are a poster child. The one exception is bipolar disease, which, even though we don't understand much about it at all, there is one thing that we do know, and we know that lithium works in about 50% of the patients, even though we don't know the target, we don't know how it works, we don't know why it works, we don't know anything about it. But if we can pry into the can and figure out what lithium's targets are, then maybe we can identify the targets and work downstream and upstream and out and figure out not only a better drug for lithium, but what the whole process is that it's modifying, the lithium response pathway, which presumably might be the disease pathway. And we figured that out. Doing that, we think, well, we didn't figure it out. We think that one of the candidates is this particular pathway that's important for cytoskeletal development, which is now, we're starting to realize is incredibly important, and that will play a big role, I think, just that whole notion over the next 5 to 10 years. But this pathway is important for dendrite formation in early development. If you knock it out, you get completely aberrant kinds of dendritic pattern, suggesting that bipolar disease and maybe other neuropsychiatric diseases are actually developmental abnormalities that might even be inherited, which goes along with what most people have a feeling, particularly if it runs in their family. Not only are their dendrites abnormal, but their dendritic density, the spine density, is absolutely abnormal compared to this, which very interestingly was just reported last month in actual brains of patients with bipolar disease coming out of the McLean's. So by working using the molecular pathway, the molecular can opener strategy, we think perhaps that what's going on in this particular disease is that this pathway, this molecule, it's called CRIM2, but that's not so important, gets phosphorylated too much, and it causes these neurite retractions, or failure of good axonal outgrowth. And what happens is it's just a little bit too high in the bipolar patients, and what lithium does is lower it. So now what we're looking for are things that lower this. In fact, if we take mice that mimic bipolar disease and we put them in this maze, the non-manic animal, the normal animal, spends lots and lots of time right in the center. The manic animal spends very, very little time in the center, and just in a manic fashion completely rotates, and spends very little time in the center. If we prevent this crimp from being phosphorylated, in other words, from being too high, and we put 2 mice in there, and we mimic manic with the manic state with methamphetamine, what we find, in fact, is that the normal animal now spends very little time in the center. In other words, he's completely manic. The animal in which this pathway is now controlled, it's as if he's been chronically on lithium. So now we think we have a target. Drug discovery is going to be what neuroscience is going to be, in the next 5 to 10 years. But what we need to do more is now take that knowledge and see if we can prevent and intervene. So let's go back to this paradigm again. We think we know the pathway. Now we think we know multiple drug targets that we could do to lower this. And now what we'll be doing is doing and finding drugs other than lithium that can lower the phosphocrimp. Now, we can do that by actually screening drugs. So drug screening and drug discovery is going to be what neurology is going to be in the future. The key, and what would be very cool, is to screen drugs that are already FDA approved, that can be repurposed, and we think we found some cancer drugs that now, repurposed, they weren't good for cancer, but they might be just perfect for lowering CRIM2. Can we anticipate, in terms of predicting, can we anticipate early on somebody who's going to get cancer? Well, I think you heard earlier today that cancer is probably a developmental problem, and that's some stuff that in our own lab we've also kind of figured out. Here's a normal neurogenic pathway in a rodent. The cells over here, the stem cells here, move out to the olfactory bulb and become neurons. If this gets perverted, and instead of doing this, the cells instead aberrantly participate in gliogenesis, not only is there diminished neurogenesis, but now they form glioblastomas. However, we know this pathway. We know how to control it. And it's mediated by the MAP kinase pathway. And in fact, if we inhibit this pathway at just the right time, we can restore neurogenesis, and now the tumor either regresses or will not form. So can we anticipate a kid, for example, who will get a tumor and intervene early? That would be the elegance of the next decade in neuroscience. Another way of doing that would be, again, in anticipating early intervention in neurologic diseases. Can we integrate, even if we know that somebody has a disease, can we integrate our cells normally, or our gene normally? We tried this with a fetal monkey of this strain. We go into the ventricle, this is in utero, by ultrasound guidance administration of neural stem cells. They go into the ventricles, they integrate into the developing brain, and now what we're able to show is that as the animal's own brains develop, the donor cells, and presumably normal cells, if these are abnormal, get integrated right within there. So it's not a cure, but maybe a disease that would be a lethal disease or an incapacitating disease is now merely a chronic disease, because now you've integrated normal, well-functioning cells, not only right in the developing cerebrum, but also you've populated the endogenous neural stem cell population. Now, a lot of these are less controversial, though I assure you that there is a lot of controversy that surrounds this.